My goal here is to better connect the idea of an ecosystem and the dynamics of an ecosystem with those of forest soils. And I want to do that by talking about the importance of forest soil carbon to the availability and turnover of nutrients in forests. Forest soil carbon is the primary reservoir of soil nutrients, especially the most limiting soil nutrients, nitrogen and phosphorus. And so understanding its dynamics helps us understand nutrient limitation in forests. Now, let's look at carbon or organic matter dynamics from the perspective of nutrient cycling. You've probably seen a schematic like this of the carbon cycle within forests or some other ecosystem. And what I want to point your attention to here is particular the transition between living organic matter and dead organic matter. This is where limiting nutrients in forests can be re-released by biological processes, and in that way determine the amount of nutrient limitation to growth of trees. Now, most decomposition is actually done by microbes. Microbes, fungi in particular, are very good at decomposing, breaking down complex organic materials. Organic material of all types is fodder for fungi, and by primarily these organisms are attacking the carbon in dead wood, in dead leaves, and so forth. And while they use nitrogen and phosphorus that is in those materials, they have the effect of a net release of those elements back into the ecosystem where they're available for growth. Now, the first thing that comes to mind with fungi may be that they are pathogens or parasitic. There are two examples here. One's a root rot pathogen. The other is actually a fungal parasite of a fungus. Pretty cool, right? Yes, they are parasites, and that does affect these processes that we were talking about. But even there, these organisms are breaking down carbon, and one of the outcomes of that can be release of limiting nutrients. Now, most decomposition is microbial, but I don't want to mislead you. Larger organisms play a role, earthworms in particular, and lots of insects break up organic matter and partially decompose it. But by and large, the breakdown of organic matter is dependent on its chemical characteristics. Look at any particular leaf or log or whatever part of a tree, and you will be looking at a complex set of organic molecules within that, and that within that part of a tree, there will be compounds that are much easier for organisms to attack and break down, and then other components that are much more difficult. For example, proteins, sugars, and lipids are easy to break down, and so once a leaf falls off a tree, those things are rapidly degraded and removed from litter material. However, cellulose and lignin are more difficult to break down, and particularly lignin is difficult to break down. And so over time, those organic matter components will accumulate and take longer to decompose. One of the interesting things about this pattern is that you can predict how fast material will decompose, and in part, how fast nutrients will be re-released from these substrates if you know the initial lignin concentration. So here's an example from California, bay laurel, tan oak, and redwood. Redwood is a relatively recalcitrant litter. It has a higher higher lignin content than bay laurel. Bay laurel decomposes more quickly, and as a result, it accumulates more nitrogen as it decomposes compared to redwood. And you might say, hey, wait a second. How can decomposing faster result in sequestration of nitrogen in decomposing material? That doesn't seem quite right. Well, what's happening there is the overall mass of that material is decomposing, is being lost much more quickly, and that results in a net accumulation of nitrogen within that material. It also results in a release of nitrogen for into the surrounding environment. So when you see fungi are pretty cool. I'm into fungi. They do all kinds of things, essential things for us, breaking down organic matter. Just nothing else in the world can do this so efficiently, particularly lignin. Fungi are really good at it, and without that, the world would be a very different place. So clearly, not only substrate, but also the organisms are important. However, there is another critical factor that influences the rate of nutrient turnover, and that's about climate. When you look across the globe, and you can see this in many different scales, as one goes from a warmer climate to a cooler climate, decomposition will slow down. And that's true even when the substrates are identical, as in this classical experiment that was done in the East Coast. You can combine both a prediction of how lignin or other recalcitrant materials will influence decomposition with climate, the overall pattern of temperature and precipitation between the equator and the poles, and you can predict how fast decomposition will occur. Now, what do those predictions say? Decomposition is rapid in the tropics, particularly when there is low lignin concentration. And as one moves further north into drier and colder climates, decomposition slows. This is pretty good. You know, in the tropics, like this example in Mexico, decomposition is rapid there. In the redwood forests, Northern California, it's fairly rapid. It's slower, but it's fairly rapid. But you get to places like the south of Spain, which are hot and dry, and decomposition starts to slow down. Likewise, when we get into the High Sierra and places like the Palisades or nearby Whitney Portal, the other photographs from Whitney Portal, decomposition starts to slow down because it's cold and it's also dry, and that is problematic. And this is just one little other thing for you, just not to ignore deserts overall, since they are woody plant communities. Something odd happens in deserts. They're extremely dry. Biological decomposition is extremely slow there. There's this surprising characteristic of lignin, which in an environment like this, which is mostly open, lignin is actually absorbs ultraviolet radiation very efficiently, and so in deserts, decomposition is actually mostly a photochemical process. Very essential there. It also results in the release of limiting